Welcome back to the studio. This is our final broadcast for this year's London Calling, and the clipboard is gone and the beer is in hand. So we're celebrating the end of 2 amazing days. I think you all know that our long-term vision as Oxford Nanopore is to enable the analysis of anything by anyone, anywhere. And this evening was about anything. We were talking about proteins, expansion of our platform so that we can look beyond DNA and RNA, epigenetic modifications, everything into the next big thing, proteins. So I've got 3 wonderful guests here to talk with me about this today. Jeff, thank you for a wonderful talk. Not only did you give a deep technical detail throughout the talk, but you also gave a wonderful introduction as to what proteins are, but why they're useful. So I wonder if you could share with the audience online why we should be excited about proteins.
Yeah, so proteins, I like to think of them as the life's active ingredients, right? So they are what primarily determine most of life's functions. So being able to understand them at the sequence level, at single molecular resolution like Nanopore enables, would be incredibly game-changing technology.
And we think about our world in sort of 4 different sectors. We think about research, we think about clinical, we think about biopharma, and we think about applied industrial uses. Winston, you work a lot in research. How do you think about proteins in the research world? What are they useful for you?
I mean, we need to know and get mechanistic insight about all these changes, all these things we're hearing about differences in DNA, differences in RNA. So what? Until we know how the proteins are interacting with them, what they result in the cell, we only know so much. We need to have the whole picture. We need to get all the way through the central dogma and understand what's happening. And for that, we need to be able to characterize protein. We need to be able to characterize the PTMs, as Jeff is showing we can do, and as Lakmal is showing we can do. So it's exciting that we're going to be able to do this soon, all from one detector.
So post-translational modifications, PTMs, what are they? What do we learn from them about biology? I'm coming back to you, Jeff. I'll come to you in a minute, sorry.
Yeah, so a lot of protein activity is regulated by these post-translational modifications. So essentially what they are is changes that happen to the proteins, to the side chains of the proteins, that modulate how that protein will interact with other proteins. So say in a signaling network, which signals will be propagated, which will not. Can also turn enzymes on and off. So yeah, PTMs essentially regulate, are sort of a switch to turn proteins on, turn them off very, very quickly. And so understanding those would give us much better insight into how biology is responding to things like disease states in real time.
So as always, all of this comes back to human health, how we can understand it more and then do things about it. Fantastic. So Lakmal, you gave an amazing talk yesterday. I was sitting in a session earlier when 5 people in a row asked you in 5 different ways on what date full protein sequencing would be available. And we haven't set a date, but I know that you're excited, and the work that you guys are all doing together is really important in this. So can you talk about the different buckets of work that we're doing to get towards protein analysis?
Yeah, so I think protein analysis, as I said, like we have, first of all, 2 different methods, short peptide sequencing and long peptide sequencing. And in short method, so we have a biomarker detection, and then we have the barcoding method. And so those 2, I think, are the first 2 applications that we are going to release as a product. Hopefully we will be releasing, or at least like work with our early access collaborators to us sometime within this year. And then the third bucket is the affinity-based methods. So in affinity-based methods, as I said, it's like indirect method, but the readout is DNA, and being DNA sequencing company, obviously we can do that. And then long read sequencing. So I think de novo sequencing is challenging. We are all working together to build the amino caller to get into de novo sequencing. So those are the buckets, basically the peptide barcode method, biomarker detection method, long peptide method, affinity method, and then towards de novo protein sequencing.
Fantastic. And you mentioned collaboration. Obviously, to get a technology like this out into users' hands takes a lot of collaboration. So Could you talk to me about your different roles in developing this technology and how you collaborate together? Winston, I know you're part of the equation.
I want to get in on it. I'm excited, but it's these guys doing all the work right now.
Yeah, I think we need to do collaborations in two different ways. First thing is to develop the technology. We need to collaborate, and then once we have something, and then to improve it further, we need to collaborate. So I think we are working with Jeff to develop the technology. That's collaboration, and we will work with Winston to improve the technology and asking questions. Why do we need this? What else we could do?
There's so many cool applications. Even people are like, oh, I want full-length de novo sequencing. Sure. But there's so much we could do right now with what is already available based on the data we're seeing from Jeff and from Lakmal. It's really exciting.
What would you be doing today?
I mean, just look at one signal transduction pathway. Look at how, we've been talking about insulin. When insulin hits its receptors, triggers this cascade inside the cell of PTMs and differences in protein levels and stuff. Right now you have to do either a series of Western blots or mass spectrometry or other ways to look at that. Man, we could just look at that all at once with Nanopore. We train that set of biomarkers and go to town with that. That's one possible thing to do. The protein barcodes, also super useful. Right now we use things like DNA oligos. Man, I don't want to switch between DNA and protein. Just put it right on the protein, trim that off, and read out the protein barcode straight out. There's lots you could do.
So what would it mean then for you to have one device with a flow cell that gives you DNA and RNA, a flow cell with DNA, a flow cell that gives you RNA, and a flow cell that gives you protein? How would you integrate that into your research programs?
For me, I would love to use that because then I can develop, I have one platform to work from to develop. And it's also really important when you think about accessibility, because if I have to buy this box to do this thing, this box to do this thing, it's like in the kitchen. You don't want a tool that does only one thing, right, when you're cooking. It's the same thing. You want to have a platform that you can use to do multiple different assays at once. So you can invest heavily in understanding and using that platform to measure lots of different molecules, especially if you're a low resource environment or, Hopkins has lots of stuff, but what if I'm somewhere else and I want to diagnose some disease or I want to test out something? I need to have a platform I can use. I mean, we know that the MinION works in space. You can't bring flow cytometers to space, but you can bring the MinION to space.
Well, we certainly can, and let's hope we can get some flow cells up to the space station soon for protein sequencing. Well, that's a really important point. We've been talking a lot about accessibility and representation in genomics over the last couple of days. And Lakmal, I know this is a particular passion of yours, and you've spent time in Sri Lanka and lots of other countries thinking about low and middle income countries and how they can improve the capacity that they have within the regions to drive their own scientific agendas. Is protein part of that?
I'm sure, so as Winston said, accessibility is a big thing. And the other thing is actually when you generate data for DNA and RNA and protein for 3 different platforms, to integrate that, there are no workflows. So if it is all 3 data types are coming from one platform, and it's much easier for us to develop workflows to understand from DNA to RNA to protein. So when it comes to low and middle income countries, obviously it's going to be, DNA RNA at the moment is huge. As I said, I keep saying, during the COVID, more than 50% of COVID sequencing in Africa happened with Nanopore, right? That is testament to what we can do with a device that is low cost and portable and affordable. So it's the proteomic space is big and it's huge, and there people can't afford mass spec, especially in low and middle income countries. If you take Sri Lanka, for example, probably there is, I don't know, maybe a couple of mass specs for the entire country. So MinION you can have in every single lab, every single university. So that will blow up completely the field.
That is it. Well, Jeff, you mentioned mass spec 1 or 2 times in your talk. Can't we just stick with mass spec? Isn't that good enough?
Well, mass spec is incredibly powerful technology, like I mentioned. It can do a lot of things. But speaking of accessibility, just mass spec is hard to access even in laboratory environments. So a lot of times a laboratory might want to study a particular protein, and utilize mass spectrometry, but they're not experts in mass spec, so they go to a core facility. So it's high cost, it's high turnaround, and oftentimes hard to analyze the data, right? So by having nanopore technology, make it much more accessible for now much wider swath of labs could do proteomics themselves.
So that's a really interesting question. So say we release a technology to the world, makes protein analysis much more accessible, people can have high yields of data. Are we ready? What do we do with the data? What other changes do we need to see in the research world to actually take that data and do useful things with it?
That's a tough question. I would say right now, it's going to be a data deluge if we have single molecule protein sequencing at scale. I think though it would be coming at a perfect time in AI to where we have now the means finally to be able to deal with these large data sets and get new insights out of them. Yeah.
What's your computational stack look like back in the lab? Do you have access to cloud, or do you do it yourselves?
Yeah, we've got our own local GPU. We also have cloud, local cloud at the university. So we've got the compute. We're ready for it. Yeah.
Fantastic, excellent. Well, look, thank you so much for the parts that all of you are playing in bringing this technology, hopefully to bring more discovery and then the development of more methods and more diagnostics and more therapeutics that can help human disease, but also agriculture, climate change, all the things that Gordon talked about on his first day. But the work that you're doing is just really high impact, and thank you for doing it, and thank you for sharing a beer. So I think I'm going to say happy end of London Calling.
Yes, thank you.
Well done.
Thank you. Cheers.
There you go. Fantastic. Well, that's the end of our broadcast for the last 2 days. We're posting all of this content online. Stay in the platform or watch our social channels over the next few days. And as Gordon said, register soon for London Calling 2026 because it sells out really fast. Have a good evening.